Hello everyone, welcome back. Today I'm here with this ninth part of this question. In this part, I will discuss some questions from plant economic botany and plant embryology. Let's start. This question is fibers associated with phloem are known as fibers जो phloem के साथ associated होते हैं उन्हें क्या कहा जाता है? Options are wood fibers, bast fibers, hard fibers और surface fibers. तो जो phloem के साथ associated fibers होते हैं उन्हें commonly कहा जाता है bast fibers. And mostly जो commercially use होते हैं fibers, these all are the phloem fibers or the bast fibers. So bast fibers may be defined as those obtained from outer cell layers of stem of various plants, and examples are flax, jute, hemp, rami, and kinef. So these all fibers are the bast fibers or phloem fibers. So fibers associated with phloem are known as bast fiber. The next examples, like we have flax, kinef, jute, hemp, sisal, abaca, banana, pineapple fiber. This is obtained from mesocarp of the coconut fruit, and the term used for this fiber is coir. Then we have wood fibers. These are the hard fibers, also known as xylem fibers. They are used for making boards or other rough items. Then surface fiber, so it is obtained from the seed surface. Like we have example cotton. So cotton is an example of surface fiber, and the term used for that fiber is known as lint. So that long fiber is known as lint, and the small fiber is known as fuzz. The next question is the place of attachment of stalk with the main body of ovule is called. Our options are raphe, hilum, placenta, or suture. So its right answer is hilum. So hilum is the structure where the funiculus joined with the main body of the ovule. So that structure is known as hilum. Hilum. So we have different parts of this ovule, like we have some attachment points. First is funicle. Funicle होता है एक stalk की तरह structure that attaches the ovule to placenta. जो placenta को attach करता है ovule के साथ. Then next is hilum. So this is the structure where funicle is attached to the ovule body. So this was the question which was asked. So place of attachment of stalk with the main body of ovule is called hilum. Next structure is raphe. So this raphe is a ridge-like structure formed where funicle fuses with the body of ovule. जहाँ पे funicle fuse होता है with the body of the ovule. The next is integuments. Integuments are covering layers that protects the developing embryo. So outer coverings are known as integuments. So ovule may be unitegmic or bitegmic on the basis of number of integuments. Unitegmic means having single integument. So bitegmic means having two integument, that is outer integument and inner integument. Next structure is nucellus. So nucellus is a mass that lies enclosed within the integuments is called nucellus. So nucellus होता है एक nutritious material which is present inside the integument. This is known as nucellus, and it acts as a reserve food material. Next is embryo sac. So this is also known as female gametophyte. This is covered by a thin membrane called as embryo sac, and it is associated with nucellus. And ovule has generally a single embryo sac formed from megaspore through reductional division, that is meiosis. Mainly the typical embryo sac consists of 7-celled 8-nucleated structure. Most of the cases 7-celled or 8-nucleated embryo sac. So 3 are the egg apparatus, 3 cells which form egg apparatus. One is egg, other 2 are synergids. Central cell that contains polar nuclei, and the antipodals, 3 antipodals. The next question is the development of embryo in Capsella, bursa, pastoris. So this is an example of typical dicot embryo development, that is Capsella, bursa, pastoris, commonly known as shepherd purse plant. So our options are crucifer type, asteroid type, solan type, or gynopodia type. So its right answer is crucifer type. In this type of embryo development, zygote divides transversely to form lower basal cell and upper terminal cell. The upper terminal cell divides by vertical division, and lower cell divided by transverse division to form 4-celled pro-embryo. Then after that they divide again and again, give rise to this globular structure of embryo. Then it will form the next stage structure, that is called heart-shaped structure. Then finally comes the torpedo stage. So these are three stages which occurs during the development of embryo. Firstly, this crucifer type of development is also known as onagrade type of or crucifer type. Here the embryo develops from the micropylar end of the embryo sac. Here zygote divides by transverse division, forming upper terminal cell and lower basal cell. This basal cell divides transversely, while the terminal cell divides vertically. Terminal cell divide vertically to form 4-celled proembryo. This structure is known as proembryo. Second division is right angle to the first division and results in formation of 4-celled stage in the terminal cell, that is called quadrant stage. Terminal cell में ही 4 cell बनते हैं and that is called quadrant stage. Then transverse division in the quadrant result in 8 cell. इसमें फिर से एक transverse division होता है that results in formation of 8-cell structure in 2 tiers, that is 4 on upper tier and 4 on lower tiers. 2-tier structure, that is called octant stage. Here again the upper tier of 4-cell of octant is called epibasal or anterior octant, and the lower tier of 4 cells constitute hypobasal or posterior octant. Here periclinal division occurs in this octant that results in formation of 6 cell stage with 8 cells in the outer and 8 cells in the inner region. Outer 8 cells present the dermatogen that will form the outer covering, and this undergoes aniclinal division that increases the number of cells, produces the epidermis, while the inner 8 cell divides by vertical division and transverse division to form outer layer of peribellum, which give rise to the cortex, central region of phloem, which form the stele. So this is how the embryo development takes place in case of dicots. Mostly in case of Capsella, bursa, pastoris, we have different types of embryo development. Like this is based upon the origin of pro-embryo. Basically 5 types का होता है embryo development. So first was crucifer type. It occurs in this Capsella, bursa, pastoris. The typical example here, basal cell plays little or no role. The next is Astra type. Here basal and terminal cells play an important role. दोनों ही cells important role play करते हैं. और जो terminal cell है of the 2-cell proembryo divides by transverse wall, basal cell plays a little or no role in the development of embryo again here. And the next is solenat type. Here basal cell usually forms suspensor cells. So जो basal cells हैं वो सिर्फ suspensor बनाते हैं. Then next is caryophyllid type. Here basal cell does division. यहाँ पे basal cell divide करते हैं. Next is chenopodial type. So both basal and terminal cells take part in development of embryo. So this is very important. So chenopodial type, both basal and terminal cells take part in development of embryo. So these are different types of. Next question, Portulaca shows fruit dehiscence of. Our options are transverse type, loculicidal type, and septicidal type or porous type. So its right answer is transverse type. Here this Portulaca shows the transverse type of dehiscence in its fruit. So fruit of Portulaca and Celosia liberate seeds through transverse rupture. The next we have porous type of seed dispersal. Here it occurs in case of poppy, that is Bower, Somniferum and Luffa. Here seeds are liberated through pores in fruits. This mechanism of seed dispersal is also known as sensor mechanism. Next is sutral type of seed dispersal. It occurs in case of pea, that is Pisum sativum, and other beans. The seeds are liberated through sutral valves. Suture के through ये जो suture है वो break होता है और seed disperse होते हैं. Next is loculicidal type of seed dispersal. Here the fruit of it occurs in case of fruit of cotton, that is Gossypium, and ladyfinger, that is Abelmuscus esculentus. The seed are liberated through the locules. Next is septicidal. It occurs in case of linseed, that is linum, and mustard. The seeds are liberated through septa or partition wall. And the false septum, which is present in case of Brassica campestris in its ovary, is called rhaplum. Next is septifragile type of fruit seed dispersal. It occurs in case of Datura and other such plant. The fruit rupture loculicidally, throwing valves away from fruit and leaving seeds attached to central axis. So there are different types of mechanisms of seed dispersal in case of different plants. The next seed dispersal and their agents through which this dispersal of seed or fruit occurs. Firstly, wind. So examples are milkweed, maple, and dandelion. The next agent is water. It occurs in case of palm tree, water lily, lotus. Animals and birds. So fruits which have pulp or juice, these are carried out by animals, like we have cherry, tomato, blackberry. And bursting mechanisms that occur in brome, jewel wheat. Next question, the effect of pollen grain on the pericarp lying outside the embryo sac is known as. Our options are xenia, metaxenia, both can be, none of the is right answer is metaxenia. So जो fruit की wall पे जो इफेक्ट होता है पोलन ग्रेन का, that is called metaxenia, and the direct effect of pollen grain on seed and fruit is known as xenia. So xenia, this word xenia was first time used by Fouquet in 1881 to demonstrate the direct effect of male parent or pollen grain on fruits or seed. For example is maize. So this xenia effect is shown in maize plant. So direct effect of pollen grain on the seed and fruit outside the embryo. Next is metaxenia. It occurs in case of date palm. So effect of pollen grain on the testa and fruit wall. So pericarp is the fruit wall. So fruit wall पे जो effect होता है pollen grain का, that is called metaxenia. So this was all about for our today's discussion about these questions. Hope people get some idea from this presentation. If you have any questions, queries, and any suggestion, give it in comment section. Thanks for watching. Have a good day.